Hello, this video is to help you with the bioinformatics tools that we're going to use to do multiple sequence alignment. For this class, I only show you the Clustal and muscle. There are many other ones, but I chose Clustal because it's historically important. But then there were other improvements, and muscle was maybe the next one. There were many other ones in between, and then even other ones that surpassed muscle. But I just wanted to show you that even if you have the same sequences and that you're comparing, so I have 3 sequences that you align using Clustal, and the same 3 sequences you align using muscle, the alignment might be different. And as I told you in class, Clustal uses progressive alignment, and the muscle will use the iterative alignment. So for the questions that I say use this approach or the other, that's when you have to realize, okay, she means Clustal or she means muscle. In real life, if you do alignments, then you are going to choose whichever tool the lab where you work uses. There's something called Tea Coffee, and I think that was probably the latest one when I was doing alignments, but I think that there have been even more ones developed. So I'm going to share my screen with you so you know where the or what the questions are. So for this first one, I'm telling you align these different sequences here, and I'm telling you I want you to do the alignment using a progressive algorithm. Not because it's better, but just because I want you to know that there are different approaches, and whenever I don't specify which one, that probably means that I ran both types and the alignment generated is the same. Okay, so when I say the progressive, I want you to look at or run Clustal. And the sequences you're comparing are this one. So this is the first sequence, as you see, is in FASTA format, which is characterized by the greater than sign, and then a description, which may be present or will be absent. And then over here, you don't see this, but there's a new line character. So the computer knows that, okay, the description of the sequence, the tag of the description or of the sequence ended, and the sequence itself starts in the next line. And it doesn't matter if there are new line characters here, it still understands that all that belongs to this first sequence. And then when it encounters, when it encounters a new greater than sign, then that's the next sequence. So I want us to see how these sequences resemble each other. So is the human kiss one closer to the mouse kiss one or to the zebrafish kiss one? So there is some gene called the kiss gene, and calling it a one, you could imagine already that there are other versions of that gene. So if you scroll down, then you'd see like, oh, there's a kiss2. And if you notice, there is the human kiss1. Let's see if there's a human kiss2. I don't think there is. Or if there is one, it's not included here. Let's see, there is, well, there is a zebrafish kiss1 and the zebrafish kiss2. So that indicates that, or you learn from experience that whenever they are numbered, then that means there's another copy of that gene within the same genome. So then you conclude that those two, the KISS1 and KISS2, are paralogs. And whenever you compare human KISS1 to zebrafish KISS1 or to mouse KISS1, you are comparing orthologs. So the orthologs are those genes that are occurring in different species. And the paralogs, they are the different versions of the gene that happen within the same species. So in that same genome, that sequence will appear twice. So just think if this was a book, then that word appears in more than one page of the book, or in more than one sentence. All right, so how do I compare them? You will open a new window, and I'm just going to type Clustal. And let me see, is it sharing? Oh, okay, so now it's, let me share with you the next window. Maybe entire screen will be best. No. Okay, then let's do tabs. All right, so I just typed Clustal, and then Google says, do you want to do the Omega version? When I was doing my PhD, it was Clustal W. So it's basically a new version of something that existed before. You could think about it as operating system. It's whatever modifications they're doing, then there will be a new version coming about. So we're going to do this one, Clustal Omega. And you see there are two choices. They both will do the same thing. I'm going to run this one on EBI. This is the European version of NCBI. So in the United States, there is the NCBI, and which houses bioinformatics data and the tools, and then the European counterpart is the EBI. So in here, it asks me, are you going to do a multiple sequence alignment of protein or DNA or RNA? And if I had written DNA and I am comparing protein, then it will flag the sequence. But if you looked at this KISS2, KISS1 sequences, you will see that there are letters that are not A, T, C, and G. So that gives you a hint that it's protein. So I just copied and pasted everything, and you don't have to worry about giving more spaces like here to tell the computer that that's another sequence, because it knows, because of the greater than sign. So because of the FASTA format, it knows when one sequence starts and the other one ends, or when it starts and ends. So then all I do, I click submit. And then right now it's doing the alignment of all of those sequences. And here's the result. So let me explain to you how you interpret an alignment. So you see it put here the zebrafish KISS2, and then there is it again here and again here. This means that the sequence for that first organism started here. Okay, then it included some gaps so that it can fit in these other amino acids that happen in this other version. So the sequence itself, if you go back, it will say MNTR and then A, L, whatever. And when it gets here to P, the remaining of the sequence continues here, E, P, whatever, until, so you keep doing that until, and so the last amino acid for the zebrafish KISS2 is a serine, is that ester. And then the frog KISS2 starts, it always starts with the methionine if you have the full sequence. That's the first amino acid from a protein. So there's, I don't even see why did they line up with the arginine, not over here, but the alignment decided it was better to put the gaps here and extend the gaps. So the second one, these are the amino acids for the sequence, and it continues here. So this order between on here the names is conserved from in each of these blocks. So then you know the top one is on the first row, on the first block is the same as the top one in here. So in an exam, I might ask you, what is the last amino acid for frog kiss 2? And then you'd write C, that's a cysteine. So that's the last amino acid in the sequence that was given here. Sometimes I ask you, what is the fifth amino acid for zebrafish kiss 2? And so the zebrafish kiss 2 is here. So first, second, third, fourth. And then sometimes people say the fifth one is a gap. Well, the fifth amino acid of that protein is not a gap. The gap was introduced to allow the alignment to happen. But in that protein, the fifth alignment is an alanine. Okay, so this is how we read the roles. What are the sequences for each of the entities we are comparing? And then here on the bottom, there are some cues, and I wanted you to figure out what do those things mean. So in some places those cues are absent, right? So like nothing here, nothing over here. And if the way you figure out what that means is you go down each column, so you see like there was no conservation here. So T, A, R, H, P, H. Okay, well, there's an H appear twice, and okay, T appeared twice, but there's a lot of variation. So since the amino acids don't agree in that spot, then no symbol was put here. So then if you compare over here, okay, what are the two dots? So then you have the L, V, and you're going to look at the amino acid table, and then you're going to see how similar are those amino acids. And then let's see, those are like the two dots. So then you have like so I is isoleucine, and then L is leucine. Then you're like, isoleucine and leucine, they sound pretty similar. Actually, they are. So then you look at the table, and you will see that on isoleucine, leucine, and then phenylalanine, okay, then that's the F. You don't have to memorize that. You can just type amino acids table, and then you will see the names. They have 3-letter names. They have 1-letter names for the amino acids. But what I want to figure out from this is, okay, what do the two dots mean? And it has to do with how much conservation there was. How about the star? So let's see, what does the star mean? In that spot, the star was isoleucine. Oh, yeah, leucine, leucine, leucine. So they're all L's. Then you know that star means something. Is it when it's only L's? Then you're like, oh, another star down here. Let's see what that means. Oh, it's all N's. And is it only when it's L's and N's? Let's see over here, another star. They're all F's. So then you're figuring out what the star means, right? And then you noticed already that when there is nothing down here, hopefully you realize that means there isn't a lot of conservation. And conservation meaning the amino acid in that position changed. Or when there is no conservation, the amino acid changed. When there is conservation, then it remained the same. When there is some conservation, but it still changed, it often changes to something that has physical chemical characteristics that are similar. So now let's see the one dot. We can look over here on this row or on this column, and then on this other column. And your job is to figure out which one is the most to the least conserved. And your choices are the star, the two dots, the one dot, or no dots. So there is a grading here of how conserved the amino acids are in each position.